Hi, my name is Mukta Palshikar. I am a third year student at the University of Rochester in the Takar lab, and today I'll be talking to you about our work building executable models of pathways using single-cell RNA-seq data. So just to outline where I'll be going with my talk today, I'll start by describing some of our recently published work that uses the Boolean model framework and bulk expression measurements to describe regulatory interactions. And then I'll move on to how we're extending that work to infer signaling modes and pathway activities from single-cell RNA-seq data. So biologists know that signal transduction pathways are not static and in fact act dynamically in concert to produce a range of phenotypic effects. For instance, this figure shows the interplay between IL-2 and IL-6 pathways. And the downstream effects of this interplay include effects such as global changes in gene expression and cell differentiation. So one way to study the context-specific regulation of these pathways is to put them together into networks or graphical models. The wiring of these networks can vary among cell types and disease states, and sometimes that's important to remember. Our lab uses a Boolean framework to think about these signal flows across networks. In these Boolean models, the regulation of a node, such as node C, is a Boolean function of its upstream nodes, in this case, nodes A and node B. A natural consequence is that these models can be simulated to generate time course trajectories, like the one you see over here, that are analogous to signal flow through the network. In addition, specific nodes in these models can be perturbed, either knocked in or knocked out to simulate the effect of internal or external stimuli. Another interesting feature of these networks is that their simulation trajectories eventually fall into limit cycles known as attractors. An attractor is a location in the state space of a network that it will move toward when simulated and to which it will return if perturbed. So you can see in this panel on the left that several states lead into the attractor state that's labeled in blue over here, but the attractor state itself has no outgoing edges that are not self-loops. These attractor states of biological networks have long been hypothesized to correspond to signaling behavior that's characteristic of specific cell types. This is familiar as the intuitive concept that distinct cell types just exhibit distinct signaling patterns. Our previously published algorithm, Bonita, or Boolean omics in network invariant time analysis, infers Boolean rules for predefined network topologies using bulk expression measurements. Briefly, Bonita uses predefined network topologies and RNA-seq data to learn and optimize regulatory rules, which are then used to perturb networks in silico. This perturbation is used to calculate a score that defines the influence of each node over the network, and these important scores, weighted by experiment-specific fold changes, are used to quantify pathway dysregulation. So while Bonita does a great job of inferring these rules from conventional RNA-seq data, we wanted to use the unique capabilities of single-cell RNA-seq data to improve our rule inference and also build a pathway analysis method for single-cell RNA-seq data. This was particularly motivated by our lab's work studying atherosclerosis in the context of HIV infection. So it's a prolonged HIV infection and treatment with CART is known to lead to both an generation of a pro-inflammatory environment characterized by changes in expression and changes in the population composition of PBMCs. For instance, the well-studied increase in the population of pro-atherogenic CD14 positive and CD16 positive monocytes. So single-cell measurements of gene expression profiles can reveal the regulatory network dynamics and the cell fate decisions that lead to these observed phenotypic changes. So our lab has recently acquired an enriched data set of singly sequenced PBMCs from 8 HIV-positive people, 4 of whom have atherosclerosis and 4 of whom do not. 16 transcriptionally distinct subpopulations were identified in this data set using modularity optimized community detection as described in these papers. The further analysis of this data set to understand the mechanics of HIV-associated atherosclerosis is really what motivated the development of our algorithm. Just to outline the single-cell bonita algorithm, single-cell measurements and pathway topologies are used to infer Boolean rules and identify dysregulated pathways. We are also currently working to use the dynamic characteristics of the inferred networks to classify singly sequenced cells into subpopulations based on their pathway activities. So beginning with the rule determination algorithm, the inputs for our algorithm are a binarized single-cell RNAseq dataset and a predefined directed network topology. These topologies are extremely useful to restrict the search space of our algorithm, and they can be obtained from either pathway databases such as KEGG and wiki pathways, although it's certainly possible to use manually built or curated networks with our algorithm. As a first pass in the rule determination algorithm, we use a genetic algorithm to perform a global search and identify a rule set that can be further refined. Candidate rule sets are optimized by simulating the network with each candidate and then scoring the match between the reachable attractor and the expression vector of each cell or sample. The underlying assumption of this procedure is that the observation of a cell represents a steady state from which it cannot easily be perturbed. The minimum error rule set that comes out of the global search is further optimized by a node-wise local search. Essentially, the regulatory rule for each node, one at a time, is modified while the other rules are held constant. And the fit of the modified rule is scored similarly to the search to the score that's used for the global search. This procedure returns a set of rules for each node that all describe the data equally well. I'll be referring to this set of equivalent rules as the ERS. We went on to test the performance of this rule determination algorithm using a set of simulated networks. The topologies of biological networks were used to generate Boolean functions, and a set of reachable attractors for each of these simulated networks was provided as a training set for single-cell bonita. So this plot shows the ability of single-cell bonita to recapitulate rules that are used to generate the training data. The height of each bar on this plot represents the mean number of true rules, the mean percent of true rules that were correctly inferred for a single network. The number of nodes in the test network are plotted on the x-axis. Error bars represent the standard error of the mean. On average, it turns out that we correctly infer rules for about 80% of the nodes in simulated networks with a range of complexity. So diving deeper into the optimization procedure, the plot on the left shows the performance of the global search for 3 representative test networks. There is a steady decline and eventual plateau in the value of the optimization function averaged over all individuals in the generation. This indicates that the fit of the minimum error rule set gets better as the optimization along with mutation and recombination progresses. The plot on the right shows that we're able to successfully narrow down the candidate rules for each node using the training data. The x-axis on this plot shows the number of equivalent rules returned by the local search for nodes with the in degrees that are plotted on the y-axis. The maximum number of possible rules increases with the increase in degree, and the better the algorithm is at learning from the training data, the smaller the ERS. We see that for the complex case of nodes with in degree 3, the average size of the ERS is less than 20. This represents a significant decrease from the maximum possible number of rules, which is 127. An inherent limitation of trying to make inferences about the network dynamics from snapshot measurements is that we occasionally run into cases where we cannot correctly identify signal flow. For instance, in this 3-node network, node C is regulated by its upstream nodes A and B. However, A can influence node C in one of 2 ways, either through this incoming edge 1 or through this route via node B. So there's no way to tell these signal flows apart unless we have time-resolved data. In order to characterize these unsolvable equivalences, we developed the total ancestor overlap score that you can see over here. Essentially, the score is the sum of the number of pairwise shared ancestors between any 2 upstream nodes. And as you can see in this panel, it turns out that the ancestry overlap score of a node correlates really well with the size of the ERS for that node. So this effectively outlines a limitation of our rule determination algorithm. We use the rule determination algorithm to infer rules for a set of biological networks using the single-cell RNA-seq data from PBMCs from HIV-positive people. Our goal remained to understand the mechanics of HIV-associated atherosclerosis at the single-cell level, which motivated the development of this pathway analysis algorithm. A perturbation-based analysis is used to quantify the structural importance of each node in the network by simulating knock-ins and knockouts in conjunction with experiment-specific fold changes from the training data. Bonita uses node impact scores to identify dysregulated pathways. A p-value is assigned by generating a contrast-specific distribution of impact scores by randomly resampling relative abundances from the data. This pathway analysis method identifies dysregulated pathways in HIV-positive people with atherosclerosis, providing an insight into mechanisms of this condition. It's important to note that as a preliminary step, we ran our rule determination and pathway analysis pipeline on the pre-classified subpopulations I described on a previous slide. So this plot shows whether or not a pathway that's shown on the y-axis over here is significantly dysregulated between cells in the subpopulation on the x-axis over here between atherosclerosis positive and atherosclerosis negative people. Pathways that are significantly dysregulated at p less than 0.5 are highlighted in red, and only pathways that are significant in at least one subpopulation are shown. As an example of some of the biological information that single-cell Bonita can provide, this figure shows a small portion of the Mtor signaling pathway, which was identified as being dysregulated between CD8 positive T cells from HIV, from atherosclerosis positive and atherosclerosis negative people. We see that Mtor over here and its upstream parent RHBB had the highest influence over the network, which is evident by the high number of outgoing edges from Mtor. Nodes without outgoing edges are assigned the lowest impact score. We've also previously shown that this impact score is not correlated with graph-theoretic measures of nodes in relativity. We hope that Bonita's single-cell Bonita's node impact score will enable experimental biologists to prioritize genes or proteins for perturbation analysis through either over or under expression. One of the most important future directions of this work is to use pathway activities to classify single cells into subpopulations. We hypothesize that incorporating network topology will more precisely identify cell type specific behavior, because we've observed that distinct subpopulations have distinct patterns of dysregulation of specific pathways. So the hypothesis of this algorithm is that cells correspond to steady states of gene regulatory networks. Just to briefly outline the algorithm, we identify reachable attractors for a set of networks for which Boolean rules have been inferred. We match observed samples or cells to these reachable attractors, as you see in this figure. A match between a cell and an attractor for the network is used as a categorical feature or attribute for downstream clustering. Our preliminary results show that it is possible to map cells, shown on the y-axis, to reachable attractors, shown on the x-axis for a network. 3 representative networks are shown over here. As a preliminary step, we also clustered cells into 3 clusters using categorical clustering algorithm. In the future, we hope to improve and expand this clustering algorithm and eventually classify large data sets based in pathway activities. In conclusion, I hope I've convinced you that Boolean models are a useful framework to think about regulatory interactions with biological networks, and that it's possible to use to infer these rules from both bulk and single cell measurements. We've also shown that these networks can be perturbed in silico to perform pathway analysis, and also we are actively working on a clustering method for single cell that uses pathway activities. That's all I have for you today. Thank you for your attention. I'm very grateful to my advisor, Julie Tagar, and all members of the Tagar Lab. Rohit Pali, in particular, is the lead author on the bulk Bonita study. I'm also grateful to Lauren, Adam, Raven, Kathy, and Alex. Our collaborators in the Margaret lab collected and processed the samples for the HIV/AIDS study. We're also grateful to Dr. Giovanni Schifito and Catherine Bunce. We're especially grateful to all participants in the HIV/AIDS study and their families, and we're also grateful for our funding. Thank you again for your attention. Thank you for listening.